What you see right now is that there's a train going on, a rolling train. People are taking soil samples and nematode samples from all over Europe. There are people investigating why certain resistant genes are no longer active at high temperatures, and they could pinpoint it almost till the amino acid level. We are investigating methylogene populations in various countries and look why they behave like they behave on basis of the genomes, so their total DNA content. In work package number 4, all of a sudden we know why certain populations are virulent while others are still avirulent. So why some of them can be stopped by resistant genes and others can no longer be stopped. It's amazing.
Well, the most exciting thing that Work Package 1 has achieved is to develop very specific molecular identification methods for the Meloidogyne species that are under investigation. Well, we're very confident that we're going to be able to achieve all of the targets for Work Package 1, particularly because we've had a fantastic season of soil sampling right across Europe, all the way from Spain across to Turkey.
So we're quite happy because we have identified molecular signatures and determinants that are very probably related to the thermosensitivity of the MI1 gene, the resistant gene. Also, we have identified cofactors that are very strong candidates to be involved in this also thermosensitivity of the resistant gene. And we also have identified some lines, some tomato lines, that look very promising to be thermostable. So I think that we have interesting results, promising results, because we have all the molecular tools that will lead us to finally found and identify these tomato genotypes that could be thermostable and with broad resistance to root knot nematodes. So we hope that we can success.
In Work Package 3, we have established the collection of virulent and avirulent populations of Meloidogyne. We have 4 species that we are working on, Arenaria, Javanica, Incognita and Lucky. And from them we have at least 15 avirulent and 13 virulent population confirmed, and still 4 not yet confirmed. So then it's something that we have to finish this year 2026. We have sent the DNA extracted of confirmed population to InRite to determine the genome and to look the differences between them. Until now, they did not get any genome yet, but there are promising results of the DNA extraction, so some have a good quality to be sequenced, and I think that in a short period of time we will have the genome. As well, at the last point of this work package, we have established a protocol of modelization of competence between avirulent and virulent populations.
For the nematode task, during the last year, we have produced several publications thanks to 2 PhD students, Océane Le Chevalier in INRAE and Arnaud Chaveling from Wageningen University. They performed 2 separate studies, and they identify the same genomic region involved in the adaptation of the nematode to the resistance factor from Solanum vernei. So it's the main contribution for the task 1. And for task 3, we have crossed a potato clone, a resistant one, with a potato clone, a susceptible one, and we have obtained offspring, descendants, and we have constructed 4 different bulk, 4 different pool of offspring, and we have sequenced those bulk and the corresponding parent. And we have received the sequence, and we have to analyze this data now. On a whole, for the work package number 4, we are very happy because the plan work is on a very good way. We have produced a lot of results, and I think that we will be able to provide the different deliverables in time.
In work package number 5, we were looking for what are called suppressive soils. Suppressive means that it is suppressing a certain pathogen. And for a long time it was a kind of a vague notion. People had the idea something like this exists, but it's hard to pinpoint. And now we all sent, let's say, candidate suppressive soils to France. They tested them all. They did a huge pot experiment, I think more than 1,000 pots. And they could confirm it. They are really suppressive. And I mean, these were soils from Slovenia, from the Netherlands. They were testing from Scotland, from France. And that is something you can only do within an EU project. In a national project, you are never able to have all these connections and to send over the borders and have a central place where everything is analyzed exactly at the same way. That's really one of the great benefits of being part of a consortium.
In Kenya, we are starting with the surveys with the farmers. In Spain and in Netherlands, we are continuing. And in Turkey, we have finished the surveys with farmers. Now, the work with the stakeholders is slightly different. As part of that work, we did the mapping exercise in Spain last year. And as a continuation of that process, we identified the beneficiaries of the 3 countries. And we were able to carry out another workshop here with local stakeholders using the information that we got from the mapping exercise last year. So year 1 gave us some information, year 2 we are applying that information, and we are going to take what we got from this local multi-actor stakeholders workshop to continue with a pathway of what are the actions we are going to take for implementation and identifying the actions that each work package has to follow for the implementation of these innovations.
So we are at the moment in the transition. Yeah, we started up, that's what you do in year number 1. You are generating results in year number 2 and also in year number 3. But now already you have a glimpse about what kind of things we are going to develop that really could help farmers or breeders or diagnostic service centers to do their job in a more precise, better, cheaper, high throughput way. I think that is where we are right now.